Hi everyone. Today our group, made up of Mariam, Taylor and myself, Justin, will be having a look at the green fluorescent protein, also known as GFP. Specifically, we will look into its history, background and development, its structure, its application in the medical field, its advantages and disadvantages, and its future. So first up, we'll have a look at and we'll try and answer a few of these basic questions. Why do we need new techniques in biotechnology? As we know, the basic aim of cell and molecular biology is to describe how events occur inside of the living cells and organisms. However, biological research in living cells has been extremely difficult due to a lack of suitable techniques. The development of a simple method to visualize cellular events in their natural environment inside of the living cell provides an extremely useful tool for a number of biotechnological applications. Using fluorescent proteins has provided this ability to visualize proteins inside of live cells. Fluorescent proteins such as GFP is a readily available, user-friendly and non-invasive technique that can be used for monitoring biological events and cell signaling. So why do we use GFP in cell biology research? Well, GFP is a naturally autofluorescent protein, which is widely used in protein tagging and in monitoring gene expression in live cells. Using GFP opened the door for the appropriate use of intact living cells as an experimental system in cell biology and biomedicine. GFP is used as a fluorescent tag to create fusion proteins with genes of interest. What is the history and development of this technique? The green fluorescent protein was found in and isolated from the jellyfish Aquia victoria, which lives in the cold waters of the Pacific Ocean. It was first discovered in 1962 by Osamu Shimomura. The original green fluorescent protein was first cloned in 1992. In 1995, by changing a single amino acid, Roger Sen engineered the first improved mutant of GFP with increased fluorescence and photostability. Since then, scientists have engineered numerous GFP variants and non-GFP proteins that have resulted in a diverse set of colours. Roger Sen, together with colleagues Martin Chalfie and Osamu Shimomura, were awarded the Nobel Prize in Chemistry in 2008. So what is the structure of GFP? Well, GFP is a protein composed of 238 amino acid residues. The unique structure of GFP contains a beta barrel with a fluorophore region inside the barrel structure. The protein is in the shape of a cylinder comprising of 11 strands of beta sheets with an alpha helix inside and short helical segments on the ends of the cylinder. The fluorophores, which are the compounds that can re-emit light upon light excitation, are protected inside the cylinders. This is referred to as the light in the can. So how does GFP give its green light? GFP exhibits a bright green fluorescent light when exposed to light in the blue to ultraviolet range. The function of the protein is reflected by its structure, in which the barrel structure shields the fluorophore from the aqueous environment, giving this region the ability to absorb light and produce a light-emitting proton. Without the barrel portion of the protein, the fluorophore would not function to emit light. Furthermore, the fluorophore region is a p-hydroxybenzylidene imidazolinone composed of glycine, tyrosine, threonine or serine, in which the serine or threonine form a unique bond with glycine to make a 5-membered ring structure. When UV light strikes the protein, the energy in the UV light gets absorbed by the bonds of this amino acid group, producing high energy protons in the green light frequency. So let's look at some of the advantages and disadvantages of GFP. The advantages of GFP include green fluorescent protons are generally non-toxic to cells. So while most other small fluorescent molecules are phototoxic, GFP, being a monomer, enables it to move readily through the cells and throughout the system without interfering with the biological processes of the organism. Their detection does not require fixation or permeabilization of cells. GFP does not require cells or proteins to become immobilized to bind to them and can freely move throughout the system without affecting a protein's life or function, which differs a lot to a lot of immunochemistry techniques that require antigens to become immobilized to attack. Its structure protects the overall formation of GFP. The tight barrel-like structure of GFP protects its formation so that when it is attached to much larger proteins, it does not affect its luminescence. It also protects the chromophore, which is the amino acid change responsible for the light emission away from solvents, so that it will pretty much be fluorescent under any condition right up until the protein is denatured. By using green fluorescent protein, and this is one of the amazing advances of fluorescent microscopy, it allows us to look into a living nervous system and see cells and parts of cells glowing as they are doing their work. And that allows us to see things that would not be possible to see otherwise. An example of this image, which was used in a photography competition run by Olympus America Incorporated, depicts the picture of the brain bow of a zebrafish after enabling 3 or 4 GFP to be produced and act as tags for different proteins throughout the fish's brain and nervous system. And as you can see, it gives us a greater view of the actual nervous system and how it's made up. So the disadvantages of GFP. Each fusion protein has to be tested for its functionality. So when you're using a green fluorescent protein, each protein has to be tested in vivo because the GFP tag is so relatively large that it could possibly affect the function of the fused protein of interest. There is no scale on which the protein can become brighter. In microscopy that uses light is limited in what it can resolve. Although light allows us to see things that are very small, there are many aspects of the nervous system that are even smaller than the wavelength of light. This can make it very difficult to resolve with standard fluorescent microscopy techniques, and for those kind of things we need to use electron microscopes, and by which and large cannot be used in living animals. Then the folding of the GFP, it's also a fairly slow process, and it can actually take up to several hours to become active. This makes the study of fast transcriptional activation processes very difficult. Now finally, let's talk about the application of GFP in the medical field and the protein's possible future applications. The application of GFP in the medical field has been about for a number of years now, having been first cloned in 1994 and then more widely recognised as an incredible important discovery in 2008. The process provides a way to see proteins and gene expression in living cells, specifically in deep tissue. This works through the binding of a GFP gene to a target gene, hence making the target gene's protein visible with the GFP bound to it. This allows us to see when a protein is created and where it can go to. According to Robert Weigart and co. and their review of GFP, imaging cell biology in live animals ready for prime time, the development of this process has given us a new understanding in the fields of neurobiology, immunology, and cancer biology. Even as early as 1995, Martin Chalfie explained that GFP can be created in many cell types and cellular compartments. and he noted the first demonstration of heterologous expression of GFP was in E. coli and the nematode Cuanahedis elegans. A methodology article written in 2002 by Douglas Sheridan and co. described ways in which functional fluorescent fused proteins could be created rapidly through a transposon-based approach, which is a class of genetic elements that can jump to different locations within a genome. As the creation of these fusion proteins using GFP had obvious medical research benefits and needed to be refined. In 2010, Yorkshire Cancer Research also wrote a paper that outlined just how important a beneficial GFP and its cousins would be, specifically in the field of cancer, with GFP being used in the medical field in an attempt to aid in the early detection and diagnosis of cancers. The paper discusses how most colours in the spectrum are rapidly absorbed. For example, when using X-rays and images within the body, such as in tissues and inside bone, they often remain invisible through these means. However, when using GFP, BFP, which is blue fluorescent protein, or RFP, the red fluorescent protein, deep images of body tissue or bone allow cancerous tumours to become visible, even in the early stages, making the cancer more easily detectable, diagnosable, and treatable. However, this paper does also state that the procedure at that point in time needed to be further developed and refined so as not to miss any small cancers in patients, and also that the specialised camera equipment needed to produce an image of adequate resolution was extremely expensive. More recently, in 2015, Robert M. Hoffman wrote a paper entitled Application of GFP Imaging in Cancer. This paper went into detail about the uses of not only GFP in finding and monitoring cancers, but also the use of other fluorescent proteins, such as red, blue, yellow, and orange. Hoffman writes that these fluorescent proteins enable the real-time visualization of the cancer cell cycle, mitosis, apoptosis, motility, invasion, metastasis, and angiogenesis, all in living creatures. Additionally, the use of the varying colours of fluorescent proteins allow the colour coding of cancer cells, helping to provide a distinction between the living host and the growing cancer. Hoffman's paper also discusses how these fluorescent proteins work in experiments, both in vivo, within the living, and ex vivo, outside of the living. For in vivo imaging, Hoffman states that the fluorescent proteins are so bright that only simple equipment, such as an LED flashlight or a mobile phone camera, are strong enough to show the protein's light. A very specific example of how this kind of imaging is done is outlined in the paper. A fluorescence light box with fibre optic lighting at approximately 490 nanometers and appropriate filters placed on top of a light box can be used to image tumours and metastasis that can be viewed with a camera to enable the images to be displayed on a monitor and digitally stored. Excitation with a narrowband filter at approximately 490 nanometers should be used. Fluorescence emission can be observed through a 520 nanometer long pass filter. A powerful handheld imaging device that inputs the image directly to a computer monitor can also be used. Additionally, the paper discusses how a variable magnification small animals imaging system containing an MT20 light source can be used for both macro and subcellular imaging in live mice, due to the imaging system having been specifically developed and holding a high light gathering capacity. In regards to ex vivo imaging, Hoffman states that metastasis could be observed in any organ at the single cell level in unprocessed tissues. in that visualization of single cancer cells in tissue is an ability beyond what standard histological techniques can achieve. So here are some pictures comparing early prototype of an in vivo imaging machine against a state-of-the-art variable magnification in vivo imaging machine. So what could be the future of GFP? The future of GFP would seem to be attempting to increase its ability to detect damaging tissues and tumours deep within the body, and refining the processes used to do this so that no cancers are missed, even when they are still extremely small. Assumedly, other critical medical uses for GFP will be researched and possibly also developed, perhaps in the fields of neurobiology and immunology. Although all research now appears to have found its focus around the GFP in the cancer field. So that's the end of our presentation today. Hopefully it's been enjoyable and educational for you. Thank you very much.